All right, here we go. We're going to start by reviewing and recapping cellular respiration, doing pretty much exactly what we did the last class period before the break. Okay, so pay attention. We're going to take it a step or two steps farther after that. Be ready for a quiz tomorrow, and for you live students, that would be on Wednesday. Okay? Cellular respiration. Aerobic cellular respiration. Here's the overall equation. Memorize this, it'll be on the final. Notice, this is glucose plus oxygen gas. We're going to have multiple steps, many different, a series of biochemical reactions. But we're going to end up with waste carbon dioxide, we'll end up with water, and we're going to end up with the all-important ATP. So that ATP is going to be essential for active transport to occur, which we've talked about. Sodium-potassium pump, which is essential for life as we know it in terms of nerve structure and function, and many, many, many other things. Okay, so the point of this process is to get ATP. Now, there's two stages of aerobic cellular respiration. The first stage is called glycolysis. So let's take a look here. Glucose is going to enter the cell, and it's going to enter the cytoplasm here. So C6H12O6. And by the way, it does that with the help of a carrier protein, and that is facilitated diffusion, which we talked about. So that's a good review sort of thing. Now, oxygen is going to come in also, but this first stage of cellular respiration is the anaerobic stage. It's called glycolysis, and glycolysis, again, is anaerobic, and glycolysis does not involve oxygen. So we're not going to talk about oxygen for the time being here, all right? Well, let's talk about glycolysis. Glycolysis literally means glucose splits. So here's glucose, and it's going to be split eventually. Well, this is a multi-step process. So let's draw arrows going down. And this molecule is going to get split in two eventually, and you're going to end up with 3 carbon molecules. And notice 2 times 3 is 6, so we still have the same number of carbons. You might be asking, where are the hydrogens and oxygens? They're here. I'm just not showing them for simplicity purposes. Okay? What's the name of this 3 carbon molecule? Well, it's pyruvic acid. All right, this signifies the end of glycolysis. But what happens along the way here? Who remembers? We're going to start off with investing 2 ATP. It's an investment. And we're going to get back 4 ATP from our investment. So the breakdown of glucose into pyruvic acid will yield 2 ATP net. And let's make sure we write net there. So that's pretty good. We get some cellular energy in the form of ATP. That ATP is going to be used to do any number of things. Next, we also are going to get a situation where NAD positive is going to grab high energy electrons from these intermediate molecules. Notice electrons are negative and that's positive, so it's fairly easy to grab electrons if you have a positive charge, because opposites attract. Okay? Not only are we going to grab high energy electrons from these, but we're also going to grab hydrogen from the local environment, and we're going to make NADH, and this is high energy. So we're going to make high energy NADH after it steals electrons from these intermediate molecules. Now, does anybody remember how many of these high energy NADHs are we going to make? What do you think?
6.
Not 6.
36.
Nope.
Oh, wait.
36 is ATP. That's over at the end. What else? What do you have in your notes? Anybody? Check. It should be from last class period. Before break. What do you have at home? How many?
2.
Yep, 2. So let's go ahead and I'm going to put 2 here, and I'm going to put 2 here. These are going to be really important. If we think of, listen here, everybody look up, listen. We can think of glucose as an energy storage molecule. That's why we call it the energy bank. We could think of ATP as energy money. When the cell needs to get something done that requires energy, it's going to spend ATP, energy money. We can think of NADH as a check. And just like if you have a check, you can cash it in to get money, NADH under certain circumstances can be cashed in to get ATP. Now, when that happens is coming up, but it's we got things to do before we talk about that. Okay. Now, remember, this is reversible. So let's put, this is glycolysis now, and we can break down molecules or build up molecules. What determines whether we break down the molecules or build up the molecules? What determines that? Well, the needs of the cell. If the cell needs ATP, then it's going to be broken down. If there's plenty of ATP, the cell might revert and take this pyruvic acid and convert it back into glucose, and then link glucoses together and make glycogen. And glycogen stores glucose for a rainy day when the cell's actually going to need it. Okay? What else is going to happen here? Each of these steps in both directions, each step requires specific enzymes. So we're going to write specific enzymes. Enzymes speed up chemical reactions, and each of these has an enzyme to speed up this process. If we eliminate just one of these enzymes, think about this. If for genetic reasons, just say that the DNA wasn't present to give the code for building that specific enzyme, let's say it's right here. If that's not present, none of this could happen. The cell wouldn't survive. Okay, that would be lethal. Lethal means deadly. It just goes to show you these enzymes are all essential for the life of the cell, and you're composed of cells, so it's essential for your life. Okay, let me get rid of that. Well, with pyruvic acid, we end glycolysis. Remember also, remember that all of this happens if oxygen is present, fine. But oxygen doesn't need to be present. This is all an anaerobic process. So I'm going to squeeze that in here. This is anaerobic, meaning no O2 required. Again, all of this is important for glycolysis. Well, at this point, we need to stop. So I'm going to draw a little stop sign. Okay. And we're going to ask a question. Is O2 present? Question mark. So is oxygen gas present? Make that is a little bit better. Well, if the answer is yes, then this will chemically react and eventually come to the mitochondria and get lots of ATP. This is the powerhouse. But if oxygen is not present, that's what we're talking about now. If there's no oxygen present, what is the fate of pyruvic acid? If the answer is no, so let's go no O2. And let's assume that you're in an animal cell, like a muscle cell. So let's say we're in an animal cell. I'll just put animal. If there's no oxygen present, this pyruvic acid is converted into a different 3-carbon molecule, and that is lactic acid. All right, let's talk about exercise and sprinting. You're running as fast as you can or as far as you can. Initially, you have plenty of oxygen. Your muscle cells have plenty of oxygen at the start. So glucose is broken down to pyruvic acid. If oxygen is present, yes. Then notice we go to the mitochondria and we get lots of ATP for your muscles to contract. By the way, your heart beating, that's a muscle contracting. And as you sprint, your skeletal muscles are contracting. So ATP is a requirement for that. But you guys know you're going to run out of breath. You're going as hard as you can. Okay? When you run out of breath, you throw your body into an anaerobic state. Anaerobic state, not aerobic. Aerobic is with oxygen. Anaerobic is without oxygen. In other words, you are using more oxygen than your respiratory system can replenish. You go into a state of what's called, literally, it's called this oxygen debt, where you can't breathe in the oxygen quick enough to replace the oxygen that's being consumed. Okay? Well, when that's the case, here's what happens. Glucose goes to pyruvic acid. Now there's no oxygen present, so no O2 present. Lactic acid builds up, and this is what causes your muscles to be sore as that lactic acid builds up. And you literally get a burning sensation. Weightlifters know about this. When you're working out hard, lifting weights, you will get the sensation of your muscles burning, and weightlifters will say, feel the burn. And that means you've got your muscles in an anaerobic state, and that burning sensation is the result of lactic acid building up. Okay? Well, if this is a bacterial cell, so let's put bacteria, let's write that in if you don't have it. Bacteria also, under this circumstance, notice, under a no oxygen state or an anaerobic state, those bacteria will produce lactic acid also. This is important in that for a number of reasons. One would be if bacteria that are surrounding and building up between your teeth, and they're secreting a concrete-like substance. It's called a bacterial plaque, so they stick there. They're not going to be swallowed. And those bacteria have plenty of food because you're eating, especially sugary stuff. They love that sugary stuff. And they're not getting much oxygen because it's really thick, and oxygen doesn't penetrate into those colonies very well at all. So notice the food that you eat is, we are in a bacterial situation now, so we're no longer in a human muscle cell. So the food that you eat is being converted to glucose. By the way, if you forgot, sucrose is table sugar, and sucrose is a double sugar, and that composes of a glucose chemically joined with a fructose. Get rid of that. And notice the bacteria can break that bond, boom, and then we've got a glucose and a fructose. So the bacteria has access to lots of glucose. Okay? Anyway, that acid builds up and it dissolves the enamel in your teeth, producing cavities. I love this analogy here because almost everybody in here, I think everybody in this class, did the egg project, transport across a membrane. We took an egg, a hard shell egg. We put it into vinegar, which is an acid, okay? And that vinegar dissolved the hard shell of the egg after 2 days, completely dissolved, leaving behind that thin membrane just underneath the surface of the egg, okay? My point here is the acid dissolves the hard calcium carbonate shell, and this acid dissolves the hard enamel, creating cavities, okay? Also, when milk sours, it sours as a result of lactic acid production. Why doesn't the milk sour inside of the refrigerator? Because temperatures are low. That keeps bacterial populations really, really low. But once it warms up, you set the milk out on the counter for 12 hours. Then the bacteria begin to double in reproductive success. So 1 divides to 2, 2 to 4, 4 to 8, 4 to 16, or 8 to 16, then to 32, then to then to 64. So those colonies of bacteria go up, up, up. The milk is thick and not allowing much oxygen in. So those bacteria, in order to grow and develop and divide, start breaking down the sugar into here and then into here, and lactic acid builds up and that curdles the milk. Of course, if you know what you're doing, you can use certain bacteria to make cottage cheese instead of disgusting sour milk. And in fact, a lot of different dairy products are utilized using bacteria and their ability to produce lactic acid. Production of cheeses, for example, is another good example. I gave the example the other day of kimchi, which is a spicy fermented cabbage. And it's the bacteria that ferments the sugar and produces the soury sort of taste. Of course, there's a lot of spices as well. So kimchi, that Korean spicy cabbage, is it's produced utilizing this biochemical process. What's it called when there's no oxygen present and we're manufacturing lactic acid going from here to here? That's called lactic acid fermentation. So let's write that. So lactic acid fermentation. This fermentation is anaerobic. So when fermentation happens, it's under a situation where there's no oxygen gas available. So whether it's making your muscles sore in a strenuous workout, or if it's causing cavities in your teeth, or milk to sour, it's this anaerobic fermentation process. Now I want questions. Questions from anybody. How about people at home? Questions. That's a good review. Why did I spend all this time? Because it's been a week since we've done this, or about a week. Okay? And why else did I spend all this time? Because it's important in terms of the life of the cell, in terms of making food products like cottage cheese or regular cheeses or kimchi, the spicy Korean cabbage. or if it's working in a bad way to produce tooth decay. Anything, any questions? Well, we're not done with fermentation, because what if we're dealing with a different living creature besides animal cells like our muscle cells, or besides bacteria? What if we're dealing with yeast? So let's go ahead and write yeast. Again, this is when there's no oxygen present. Everything on this side is no O2. So everything on this side of the stop sign, there's no oxygen present. So it's, the key word is anaerobic. Well, you're familiar with yeast. If you aren't, yeast is present in, baker's yeast is essential for bread to rise, where you add the baker's yeast and the yeast will release carbon dioxide and make that bread nice and airy and a little bit fluffy, as opposed to non-leavened bread, which is thick and doesn't have that air content that we associate with normal bread. But what if we take sugar in the form of fructose, like from a ripe grape? You with me? And what if we crush up those grapes? And on the surface of those grapes, naturally accumulating, like out in the field now, the surface of the grapes will naturally accumulate yeast. With me here? So we don't even have to add yeast from an outside source for what I'm talking about here, because the yeast are present on the skin of the grapes, okay? Now let's take those. Let's crush them. Let's seal. Make it an anaerobic environment. We don't want any air getting in. So you with me here? Might want to use a barrel for this. Seal it. No oxygen getting in. Those yeast will need to survive. They'll need to make some ATP. So what they're going to do is take the sugar, like fructose. Fructose has this same chemical formula. Okay, they'll take the sugar, convert it into glucose in one quick step, and then the glucose is converted to pyruvic acid, and then the yeast are going to ferment the pyruvic acid into ethyl alcohol. So let's go ahead and write ethyl alcohol. And that is a C2 molecule. So that is a C2 molecule. Of course, you start with 2 C3s. Now we're going to have 2 C2 molecules. Notice we've lost a carbon. This alcohol is going to be produced, but something else, a gas that you're very familiar with, because remember, matter is neither created nor destroyed, law of conservation of matter. If you lost carbons, they didn't disappear. They just left as a gas. What famous gas are we familiar with? That's the end product of many biological processes. Yeah, it's carbon dioxide. Thank you. So we're also going to get plus 2 CO2 molecules, gas. So notice, if we take yeast, we give them food, we prevent oxygen from entering the system here. The yeast will break down the sugar. The sugars were originally in the grapes, right? And they'll convert the pyruvic acid into ethyl alcohol. Make a note that the ethyl alcohol is the alcohol that's in beer and wine and spirits. Okay? So make a note, ethyl alcohol. That's the alcohol in beer and wine and spirits. And this is alcoholic fermentation. And this is how beer and wine production occurs. Notice it occurs in yeast. You're not getting alcohol fermentation in animal cells like muscle cells. Okay? So what's happening here is that the yeast do the work. And then the companies that sell the beer and the wine, granted, there's an art to this, and you got to know exactly what you're doing for anybody to buy this stuff. But it's really the yeast are doing the real work, right? It's the yeast that are making the alcohol from the sugar. Okay, and the people that work at the distillery has the alcoholic fermentation going on in barrels or vats or whatever. Okay. Questions now. We've got lactic acid fermentation, which occurs in animal cells like muscle cells. When your muscles are deprived of oxygen, lactic acid builds up. Bacteria are going to produce lactic acid from sugars also, if there's no oxygen present. Notice yeast will ultimately they convert sugar into ethyl alcohol and carbon dioxide. Make a note that the carbon dioxide is the fizz that's in champagne. It's the fizz that's in beer. And it's being produced by this process. So make a note of that off to the side. I'm out of space here. All right, I want you to be aware, if you open up a soda, Pepsi, it's pressurized with carbon dioxide. With me? That's the pop that you hear. And then you see the bubbles rise. That carbon dioxide is pumped in at the beginning of the process of making the soda. So the CO2 was pumped in from an outside source. On the other hand, champagne, which pops and bubbles and fizzes, right? Champagne releases lots of carbon dioxide, but the carbon dioxide was made biochemically. The carbon dioxide's not pumped in from the outside world. There's a big difference between the two. And of course, we make carbon dioxide biochemically also. And notice this reaction up here. Notice the CO2 that's produced is going to be produced with the presence of oxygen. Okay, so you got to have oxygen for all of this stuff to be produced. By the way, does lactic acid fermentation in your muscles, does that produce carbon dioxide? Look around here. Do you see any CO2 being produced as you get to there? Do you see any CO2 here? No. So when your muscles ferment lactic acid, no carbon dioxide is produced. For carbon dioxide to be produced, you need what? You need oxygen gas, and that's the next stage. This was stage 1. Glycolysis is stage 1. And stage 2 is the aerobic stage, where oxygen gas is present. And when oxygen gas is present, we'll produce these products. The mitochondria is going to be really, really important now, and we're going to get large amounts of ATP. As opposed to, if we go to lactic acid, how much ATP do we get? Look around. Isn't it 2 ATP net going from here to there? Fair enough. So when oxygen is present, boy, you get much more you get much more bang for your buck being sugar, glucose. Lots more ATP if oxygen is present. What happens here is a important process, so important it's essential for life, but we've run out of time. So that will be our next lecture topic. Y'all out there at home, make sure you review everything we've done with energy, potential energy, etc. Everything we've done with these processes going on here with glycolysis, lactic acid fermentation, ethyl alcohol, and alcoholic fermentation. Any questions at home? Questions at home? Anything out there? You guys, really important quiz tomorrow. I'm thinking test in here is going to be Thursday, Friday. Test, you guys hear that? Test, Thursday, Friday.